After a first degree at University College London, Gillian Griffiths moved in 1980 to the LMB for her PhD, supervised by César Milstein. Her project produced exciting insights into the process of somatic hypermutation, the way that B cells tailor their antibody genes to hone the immune response to pathogens. Today, Jillian is Professor of Cell Biology and Immunology at Cambridge University. Her lab blends these two fields to investigate how T cells recognise and kill virally infected and cancer cells, using insights gained from genetic diseases and parallels between different biological systems to identify the molecular mechanisms underlying this process. Gillian is a Fellow of the Academy of Medical Sciences and a member of the European Molecular Biology Organisation, and in 2013 was elected as a Fellow of the Royal Society. Welcome, Gillian.
Thank you.
You first encountered César Milstein at a rather challenging PhD interview, I think.
I did indeed, yes. I had been visiting various labs where I had been invited for interview, having written letters to individual PIs, which is what you did in those days. And César, I knew was an immunologist in Cambridge, but I'm afraid my detailed knowledge of everything he'd done was perhaps rather less than it might have been. And my mentors at University College had said, well, he's quite famous, so don't be surprised if he can't take you. So with some sort of trepidation, I visited the LMB. César was, of course, very nice, but César was typically César. And so you would have a conversation with him, and suddenly he'd stop and go, yeah. And there'd be a long silence. And you wondered, what am I supposed to do? Anyway, we had a very nice discussion about what I was interested in. And at the end, he said, well, I'll be interviewing lots of students, and perhaps I'll take the one with the best degree. But you'll be looking at lots of PhD places. So could you tell me could you write to me and tell me where I am on the list of PhD places you've applied for? And so I said, okay, and left. And I went back to UCL and went to my professors and said, what am I supposed to do? This is very strange. So Martin Raff, who was one of the immunology professors there, said to me, oh, that's easy, Jillian. You just say, I'll tell you where you are on my list as long as you tell me where I am on your list. And that did the trick. He wrote back saying, you're first on my list.
Well, that was handy.
It was handy, but actually in hindsight, I think it's actually a very clever thing to do, because I found that the people that you really want to come to your lab are the people who really want to come, who are determined. So I think actually the usual César insight.
Yeah. And this time was when you first came across the divisional secretary in PNAC.
Oh, Peggy Dowding.
Yes.
The wonderful Peggy Dowding. She probably wrote that letter, actually.
Yeah.
I wanted to bring a copy of it, but I've mislaid it at the moment. Yeah.
So tell me about Peggy.
Peggy was wonderful. So Peggy would... she really organised César at the time. I guess actually was Fred who was the head of PNAC at the time, but she knew how things worked, and she was very caring towards the students. And if you ever had a problem, then she would tell you what to do. Very friendly. And the best thing that she ever did was during my PhD viva, when I guess we probably started at 2:00 and it got to 4:00. And she decided that it was time to end. And she came in with a cup of tea for everyone and said, well, I think it's tea time now. Time to stop. So that was a very typical Peggy story.
Yeah. I read somewhere that she used to rewrite all César's papers and speeches so that he sounded a bit more fluent than he was in real life.
That would make a lot of sense. I'm sure she wrote the famous letter to me.
Yeah. What was the Millstein lab like then when you arrived?
I thought it was very friendly. For me, it was quite incredible. I'd grown up in the southeast of England, in a place which I think would lack diversity, would be a way you'd phrase it today. Mm. And when I came to the whole LMB, actually, it was incredibly international and diverse, and that certainly was true in César's lab. He had a long-standing collaboration with Olli Makkala, who was, of course, Finnish, and Matti Kaartinen was there working away, who was incredibly Finnish. And then there were, of course, people from South America, lots of people speaking Spanish. But César was very clear that we were in England and that if there was one English person in the room, then English should be spoken in the lab. So it was warm, it was friendly. There was a wonderful Italian, Giovanni Galfrey, who would make gnocchi, fresh gnocchi on a Saturday afternoon, and then he would invite everybody over for dinner. And other days, César, who loved to cook, would go home every lunchtime to cook his lunch. And the weekend, again, he would often cook paella and invite different lab members around. So it was like a whole new world to me, coming from this very conservative town.
Was there an expectation you were going to have to work really hard?
I think everybody wanted to work really hard, was my memory of it, that you wanted to make your experiments work. And César, when I started, set me off in experiments that were quite different. So I remember there was one thing where he wanted to try a new way of screening for antibodies, and it involved, it came from George Church, actually, in Wally Gilt's lab, and it was a very odd technique where you ended up inverting a plate with a filter on it, and you wanted red blood cells to stick to your antibody where it had been secreted. You had to do it under a bucket of water. I didn't get it to work. But an expectation that you had to work hard, I mean, I don't remember. The expectation came from within, unless you're counting David Bentley, who we'd all go out, we'd run our gels and we'd go to a movie. And then initially I thought, well, after the movie you went home. And he looked at me and said, hey, party's in the lab. You go back to the lab.
So everybody's social life revolved around the lab and being in the lab then, yeah.
Yeah, I'm sure you remember that.
I do, yes, I do. And so César was, of course, half of a partnership with his wife Celia, who I'd really like to talk about with you because I think she was an amazing person. So when did you first meet Celia?
Oh, gosh. Well, she was actually working in the lab for some part of when I was there. And I think not initially, but of course, they were a lovely couple. They would invite you over to their house. They would invite you. I remember going out cycling. Maybe it was going cycling to Mark Brescher's house, and we all they had a tandem that they used to ride. And
really?
Yes.
Oh.
No, I can't imagine riding a tandem with César, but anyway, Celia did. So Celia was so she was in Babraham to start with, but she would frequently come to the lab because she would want to learn molecular biology techniques that she would then transfer to Babraham. They were very much a couple. They would do everything together. I remember one time they went to Spain to one March meeting, and they came back with a poster, a picture of a sort of classical painting. They said, this portrait of a woman reminded us of you, so we thought we'd bring it back for you. So, yeah.
Yeah. So Celia and César basically looked after the immunologists.
They pretty much adopted us in a very gentle way. Not that you felt that they were overwhelming or prying into your life, but it was just you were adopted into their sort of social life. And César was very generous. He was always taking us all out to dinner, and he'd never let the students pay. So it was, you felt like a scientific family. I think of her, certainly in hindsight, as my scientific mother. And I miss her enormously.
Yes. And of course you were there during the Falklands War.
Yes.
What's that like?
It was, it wasn't so bad for me, but it was pretty bad for some of the people working in the lab. So Oscar Baroni, who was an Argentinian, had his funds frozen completely. And during that time, Cesar and Celia would, of course, lend the money. So he and Teresa, and they had 2 young sons. But that was typical of Cesar. I remember Celia at that time telling us all a story about how, when she was growing up, that in the comics, down the bottom of the comics, it was, say, Los Malvinos sont argentinos, which is probably my very bad Spanish saying, the Falklands belonged to Argentina.
Even on the children's comics?
That's what she told us, yeah.
Gosh.
That it was sort of, yeah, you were indoctrinated with the idea that these little islands were definitely Argentinian.
I gather when you were a PhD student, you also had a little bit of unwanted attention from various lab members. Is that correct?
Oh, well, not so much lab members. I mean, I was a bit of a novelty because there were very few PhD students. I think only 4 of us in our year, and only one girl. And it didn't really matter that you were a girl, at least as far as I could tell, because the pressure was on all of us to produce the best science. You wanted to be the one who did the best experiments. But you know, you got a lot of attention from the other students, and so, for example, Gerard Evan would come by. His lab was next door to mine, and every tea time and coffee time he would be passing by the door, and my bench was right by the door because I was new, and he would always stop to chat, to the point where I went to Cesar and said, "Cesar, could I have a bench away from the door, please? Because Gerard keeps distracting me from my sequencing." Of course, Gerard and I are very good friends.
Yes, just as well. Yeah. And you were in a Christmas party skit, which covered some of this material, right?
Yes, indeed. So there was a very good social life with lots of very friendly sort of Americans. We were always playing charades, and there was a sort of running joke that some of our colleagues from across the Atlantic liked to talk a lot and were quite proud of everything they did. And so Gerard, of course, was organising the Christmas parties, and he decided we needed some intermissions. And so he dragged me in to become one of the adverts where I was meant to look very dejected. And the voiceover was, "Are you tired of Americans always talking?" And then would come, "Try new Yankaway." And there was a spray can that appeared, which I had to spray over Kevin Stroll, who was sitting there initially yammering away, and appeared some tape across his mouth. The Yankaway worked. I'd love to find that movie. I don't know where it's gone to.
Yeah, that would be fun,
It
wouldn't
would be
it?
good, yeah.
So let's move on and talk about something slightly more serious, which is your somatic hypermutation project. Yeah. Can you tell me something about that? Maybe first of all explaining briefly what somatic hypermutation is.
Yes, so within the variable regions of the genes encoding antibodies, there can be point mutations, and the project really was to look at how point mutations evolved during an immune response, and whether that had an impact on how good those antibodies were, what their affinity was for their target.
So these point mutations only arise after the immune response begins?
Well, yes, but that wasn't really known. It was known that there were variations in the sequences of variable regions, but it wasn't known sort of how they arose or when they arose. And so what César had very cleverly done is he'd taken a very defined antibody response against the haptenoxacillone, which he had done in collaboration with Olli Mäkelä in Finland. And he decided to immunize and make monoclonal antibodies and use them in this case to find what does the immune response look at day 0, day 7, day 14, and how do those V regions change. And so my job was to try and sequence the V regions. So at the time, many other labs were doing this across the States, but they were using protein sequencing, and you couldn't get very far with protein sequencing. And so what César had suggested was that we should use a technique developed by George Brownlee and Pamela Hamlin, and which was to sequence the messenger RNA. And so it was dideoxy sequencing using reverse transcriptase, and you could then sequence and see. Of course, the gels were nowhere near as beautiful as the DNA gels coming out from all the labs around me. Mm-hmm. So, but nevertheless, they were very good because you could read the entire variable region with different primers. Mm-hmm. And so I was doing this, and I could find that there were little mutations appearing at day 7 and then more at day 14. And what César was doing, because he wanted to do lab work at that time, was he was taking the purified antibodies and measuring the affinity of the different monoclonals where we found the different point mutations. And so between us, we showed that it was somatic hypermutation that was giving rise to the affinity maturation. You could pinpoint particularly mutations that really increase the affinity. So, yeah.
So this sounds amazing, but I gather that there were some people in the lab who didn't think this was such a great project to be doing.
Well, there were various reasons for that. There was Kevin Strahl, who always thought that our DNA gels were really silly and that anybody could do it. And of course, mine were RNA. He was very nice. He didn't tease me about the RNA gels. But there was always sort of this competition, really, to see who could have the most beautiful and read the longest and the furthest and so on. So that was one aspect of that. But there were also people who would come and ask me, what do you work on? I said, well, I'm looking at point mutations in variable region genes. Why are you doing that? And I said, he said, everybody else is doing that. Lots of labs in America are doing that. And that really, there was no internet. So I went to Cesar and I said, Cesar, apparently lots of labs are doing this. And he had this big smile that came over his face when he knew he was right. And said, but they're not doing it properly. And what he meant, of course, was that they weren't sequencing. They were just getting a little bit of sequence using protein sequencing, and that they and they hadn't set up the system to look at the timing of day 0, day 7, day 14.
So they weren't watching maturation happening?
No, they couldn't do it, right? So he'd set up the experiment in such a way that you could really see the details and you could see the order of events. And it's something I find I still do now. I mean, I'm looking at killer cells, but I'm working out how the different events are ordered because it tells you a lot about the mechanism. So yes, I can still hear his voice in the back of my head. I say this all the time, whenever I'm setting up a different set of experiments, I can still hear, but they're not doing it properly. They must do it properly.
When did you realise that what you were doing was really quite important then?
So that was the very first conference I went to was in the south of France, near Marseille, and it was a conference on idiotypes, or idiot types, as Michael Neuberger liked to call it. And I went and I was giving a talk because I don't know whether Cesar didn't want to give the talk, so he suggested that I gave the talk. And as soon as I got there and I started hearing the other people presenting their work, I said, but, they've got 20 amino acids. How can they see mutations? And that's when I realized what Cesar had meant. Yeah, it was really interesting.
Yeah. And what was the reaction to your talk?
So the reaction to my talk was Cesar had told me that when I went to a conference, I should learn what not to say. And I thought, what is he talking about? I don't understand. But when he went and we decided what I was going to talk about, I had my slides, and it was evident that there were some mutations coming. But of course, everybody then was very excited, and they wanted to know what the mutations were and what we were getting at the later day, because I think I just presented the day 7 results at that particular time. So, but there was a tangible excitement, and once you get that bug, it stays with you. Yeah. It was really fun. Huh. Yeah.
So he allowed you to present the 7-day data, but held the rest back for suspense reasons? Or?
Well, I don't know, actually. It was partly because it wasn't finished, and partly, I think, for the suspense. I think he had a flair for drama, Cesar. Yeah.
And, of course, you were in Cesar's lab when he won the Nobel Prize.
I did indeed.
And how was that?
That was great. So in fact, the first year I was there, I think there were people going around saying, it'll be Cesar's turn. I said, Cesar's turn for what? Because you arrive in October, which is more or less Nobel Prize time. Yeah. And they take the prize. And I thought, what prize? And then, of course, there was Fred got his second prize.
So he got his second prize the year you arrived? Is that?
Gosh, now, was it 80 or 81, Fred's second prize?
I don't know.
Yeah.
Let's say 80.
Let's say it was very soon after I arrived, and then not long after that there was Aaron Klug, who I remember decided to buy himself a new bicycle, and I met him in the bike sheds after he got this new bicycle. He was looking at it and he was really stuck because he couldn't work out how to unlock the new bicycle. So he may have had a Nobel Prize. Yes. And then then came 84, which was the year as I was finishing my PhD. And that's when César got the Nobel Prize. And so the best story about that is related to the picture I brought along.
So can you show us what you brought along?
So I brought along a picture of, so it actually has Cesar and Celia and John Jarvis, who worked with César for many years. And this is the very first bottle of champagne after César heard that he got the Nobel Prize. And this bottle of champagne had been stored away by Franco Calabi in the lab. And Michael Neuberger, pictured here, had said to Franco, "Here, we need to take this champagne because César has got the prize." And Franco's response apparently was, "But I was saving that for something really special." I get so confident that César would get the prize. But anyway, this is a great picture. And I keep this by my desk even now because César was, one of his mottos that he taught me was celebrate before it's too late, which meant in those days that you should celebrate when you send the paper off and not when you get the reviewer comments back. But in his case, there were plenty to celebrate.
Yeah, absolutely. So did he change at all after he won the prize?
He did change a little bit because, whenever you'd asked César something before, then usually it was, no, that's not right. And he would pull down, he had all his papers in folders above my bench, actually, and he would pull them down and say, no, I showed in such and such a year. But then I noticed when he got the prize, suddenly it was, yes. I don't know whether he realised that.
So he had a bit less to prove after he won a Nobel Prize then?
I guess, yeah.
So what are the things that you learnt from your time here and your time with César then?
Well, gosh, I learnt so much, actually. And in hindsight, the more I look back, the more I realise how much I learnt from César. I learnt the excitement of doing research. I learnt the importance of looking at the details, and because all the interesting stuff's in the details, and many people rush ahead and they miss the really intriguing insights, and that's something that certainly played out in my own research in my own lab. I learnt that science can be a wonderful community, and that you really find your friends over many years in science. And I learnt about, it was like the first view I had of a big outside world with many people from many different communities and different countries. And I mean, it was wonderful.
And you also learnt, I guess, never to have your bench by the door of the lab.
Absolutely, never by the door. At least not if Gerard was around.
Gillian Griffiths, thank you so much for coming to talk to us today. It's been great.
My pleasure. Take care.